All right, guys, so we're going to do chapter 9 notes today. We're going to do sections 1 and 2 together, and then another day this week we'll do sections 3 and 4, and there might be a small fifth one. So we're going to talk about the cell cycle and cellular reproduction. You already know about this because you had to do a hyperdoc on it. So it's almost like a flipped classroom with this chapter because you should already know what I'm talking about. So it should be easier for you to do your shorthanded notes and to put your notes in your own words because you're already familiar with this information. So 9.1 is the cell cycle. These notes are super simplified. They're not, if you read the book, read the textbook, which you should, especially if you're not making great grades, it's a lot more in depth in the book. So what is the cell cycle exactly? It's an orderly set of stages that takes place between the time a eukaryotic cell divides and the time the resulting daughter cell also divides. So if we think about it as far as our lives, and you make an analogy to humans, so it says when one cell divides to the time its daughter cells divide. So that means from the time your parents started having children to when you started having children. Okay, that's what essentially what it means. When it starts to divide, and then when its babies start to divide. That's a cell cycle, or you can think of it as a quote life cycle. So this is what you should have drawn yesterday. You have G1, which is your growth phase. You have S phase, or your synthesis phase, which is where your DNA replicates. We're going to go over all this. And then you have your G2 where you grow a little bit more and you get ready for cell division. So this is where you're going to start making like double organelles and stuff, getting ready to pull apart. All three of those are in a thing called interphase. You spend a ton of time, your cells spend a ton of time in interphase. And there's also some checkpoints that we're going to go over too. Checkpoints are like roadblocks to make sure everything is going smoothly. And if it's not, then it's going to, dump it out. So like I said, interphase is where cells spend the most of their time. It's around 90% of a cell's living time, I guess you should say. During this phase, cells perform their usual function. So they're doing their job, okay? Whether their job is, if it's smooth ER in the liver, and they're, it's an alcoholic body, and they're just constantly, detoxing that person from alcohol. It could be, a nucleus. It could be whatever its job is. It's just doing its normal thing. Like I said, it consists of 3 stages. It's got G1, which is growth, for the first growth, or growth 1. S stands for synthesis, and G2 stands for growth 2. So the G1 stage is where the cell grows and is constantly performing daily functions. So whatever that cell's job is, it's just getting bigger and doing its duty for your body, or for the organism's body. So fun fact, I always tell my classes, especially when I'm in there, I get off on a tangent about my mom, but nerve cells and muscle cells typically do not replicate and divide. So they enter a phase called G0 phase after interphase. And so they don't go through a cycle, like it's like one and done. It's almost like if you think of somebody, staying with our analogy to humans, it's somebody that does not have children, basically. So if something happens to that person, or tragically happens to that person, we're thinking of them as cells here. They're no more. They can't replicate anymore and make more. Their genes are not being passed down. So if people that are paralyzed, that's what paralyzation is. The nerve cells in your spine don't heal themselves. They don't replicate. So once you damage those cells, it's over, right? That's why you have paralysis. My mom, I don't know if you guys ever noticed, but she has drop foot. So when I was in 9th grade, we had a horrible wreck. She got kind of pushed up we hit a tree, and it was after a basketball game. It was December the 17th or 18th. But we were coming back home. We lived in Little Coon, and we hit a tree, and her seat didn't the function of like moving it back and forth didn't work right, so it slung her in the dash, and her foot got caught up under the seat. So all of her nerves, her sciatic nerve got damaged down her left leg, and she can't pick up her toes or her foot. She has no control over it because that wreck damaged those nerve cells, and her body cannot make those back. So she has to wear a brace when she's wearing flat shoes. You'll never see, Miss Hicks, which that's my mom, if you guys didn't know that. You'll never see Miss Hicks because she doesn't want to show her brace. But I can't tell, but you might can tell. But I think that's cool that, I can use her as an example and kind of get your attention with that. So the next stage of the cell cycle is the S stage. This is where DNA synthesis occurs. So when you synthesize something, you're making a copy, or you're replicating something. You're making something, okay? So this is where there's a whole chapter on DNA replication, which I love. I'll probably come teach you some of those days if the COVID numbers go down. But you have a thing called chromatids, which is each double helix of DNA. And then you have another definition in this stage called sister chromatids, which is two identical chromatids. This is after you replicate them, and they're called sisters. Why? I don't know. Everything is called sisters and daughters. I never understand it, because most scientists at this time were men, but they named everything after women. People name boats after women too. All right, so G2 stage is the last stage or phase. Did I say? Your book says stage, but sometimes I'll say phase. I'm sorry if that messes you up. But in interphase, this is the last one. This is where the cell synthesizes the proteins that will assist in cell division. So it's prepping to have a baby, okay? This is the prep. So when I say on your test, when I say what's G2, you're going to say prep for nuclear division. You're prepping for the nucleus to divide. So remember, that's the powerhouse of the cell, not the powerhouse, that's the brain of the cell. I always want to say powerhouse because your DNA is literally everything, or the cell's DNA it holds the information for all. So it's like your brain. I always want to say power because I think about something really important, but that's the mitochondria. Okay, so this is where we're going to make proteins for the microtubules, and I'll probably do another Edpuzzle because some of you bombed that one, which is ridiculous, and make you watch one about mitosis, about microtubules. So then the last stage, which is not included in interphase, is called the M stage. So we're just lightly touching all these, and then we're going to go a little bit deeper in a minute. So mitosis is the M stage, or part of it. Some books will include these two things, and some books will just say the M stage is mitosis and leave cytokinesis out of it, but it's part of it. So mitosis is nuclear division. This is actually where you recreate the cell duplicates its nucleus, okay? So daughter chromosomes are distributed by the mitotic spindle to two daughter nuclei. So this is where you have to line up all of those chromosomes for that organism in a perfect line, and you have to pull them apart correctly. If you don't pull them apart correctly, you could have a cell that doesn't function correctly, and the organism has to almost abort the mission, I guess you could say. Or you might have some sort of a genetic disease or genetic disorder from this. So this is really important. And after you pull those chromosomes apart, you have a thing called cytokinesis. So this is really easy to remember because after I have one cell, and now I've went through mitosis and I have two nuclei, okay? I have to split the cytoplasm. So how I do that is a thing called cytokinesis. It's super easy to remember: cytoplasm, cytokinesis. This happens after mitosis, and it's the division of the gel-like substance called cytoplasm of the cell. So you have things that control the cell, okay? You have growth factors, which are signaling proteins received at the plasma membrane. These signals ensure cell cycle stages follow one another in a normal sequence. It just makes sure everything is working correctly. So you have these checkpoints because of this. And these prevent cancer more than we all realize in all organisms. So a weird thing too is when we think of cancer, we think of us, right? We're super self-centered. I always think of us when I'm talking about cells too. But all other organisms have cancer too, because what cancer is, it's just your cells attacking yourself. It's just, I mean, that's why it's so hard to kill. But all organisms do have cancer. We just don't know it, or I guess care enough about it to, do anything about it. But we would have a whole lot more cancer if it wasn't for checkpoints. Okay, there's 3 in the cell cycle. You have these things called cyclins or cyclins. Cyclins increase and decrease as the cell cycle continues, and these have to be present for the cell to proceed in the cycle. So if they're if they decrease, if the cell checkpoint finds something, it'll take the cyclins away, and it won't let it move forward anymore. So you can see that where the red is, these are where these checkpoints are. Okay, I forgot to put on your thing yesterday the M checkpoint. All right, so the first checkpoint is the main checkpoint. So let's look on this diagram. So the yellow, this is where I just grow and I just do whatever function I'm supposed to do as a cell. So this G1 checkpoint checks my original DNA to make sure that I'm okay. This is almost like checking your genes before you have a child to make sure that you're not carrying any genetic disorders, that you're super healthy. If the DNA is damaged, a thing called apoptosis will occur. Apoptosis is programmed cell death. It's like cell suicide, okay? The cell, your body is super efficient, or an organism's body is super efficient. So it's going to remove the cells that aren't working correctly or that could potentially cause damage to the organism. And for that, we should be super thankful. So the G2 checkpoint is, so we go through, we grow in the yellow. In the S, we double our DNA, okay? And we get to G2. So G2, so remember in S, we have 2 sets of DNA. So guess what we have to check now? We have to check that we replicated it correctly, because you can make a ton of mistakes when you replicate something. So this G2 checkpoint makes sure that the DNA that you replicated is perfect. If it's not perfect, guess what happens? Programmed cell death happens, which is called apoptosis. Okay, these are like little stop signs in the road, and it says stop, let me check you. And then you go through something else and it's stop, let me check you. If you're not good, get out of the way, go to the trash. So the last checkpoint, which I forgot to tell you about yesterday, is called the M checkpoint. This is where the spindle assembly, this is called the spindle assembly checkpoint. And mitosis will not continue if chromosomes are not properly aligned. So let's look back. This is in the green, which is where the nucleus divides. So if these chromosomes aren't lined up correctly in the middle of the cell, or in the middle of the nucleus, and the microtubules aren't attached correctly, apoptosis will occur. And sometimes, we see from Down syndrome, because you're missing a cell. You're missing a chromosome. This baby's getting to me, y'all. You're missing a chromosome. So sometimes things do slip through, but our bodies do a great job, and organisms process does a great job of preventing the majority of mistakes. So apoptosis will occur if they don't line up properly. So what is apoptosis? You need to write down or know that it's programmed cell death, or you can put cell suicide if you want. It's gonna kill itself for the greater good. Cells round up and get blisters. So they kind of swell up and they get little blisters on them, and cell fragments are engulfed by white blood cells or neighboring cells. And what happens is those parts are just reused. So your body, or an organism's body, is super, super efficient. Nothing goes to waste. I can never. I'm terrible at pronouncing things. I think you pronounce this caspases. These are always present in the cell, but they're inhibited, so they're kind of turned off. And when the switch is turned on, this will bring about apoptosis. So, like, think about the stop sign happening, and them saying, "Let me check your DNA," and your DNA is something's wrong with your DNA. So their little switch or little button happens, and that little policeman with the stop sign pushes the button, and the Grim Reapers almost like come and get you. That's what these things are. So apoptosis keeps the body in balance, and it maintains homeostasis. If we didn't have apoptosis, we would have so much, our bodies couldn't function. In human embryos, fingers and toes, they're originally webbed like a duck, but when most babies are born, their fingers and toes aren't webbed anymore because of apoptosis. So look at your hand and spread your fingers out. You can almost see where they were webbed. Like I can kind of touch mine and my skin. Those were like almost to your fingertips. But because those skin cells and those tissue cells in between your fingers, when we were embryos, I guess you would say, killed themselves. They had programmed cell deaths for the greater good so that we could have fingers and toes. Cells that are also infected with a virus go through apoptosis. So think about those that have COVID right now or that have had COVID. Your body tries to help you get rid of that virus quicker by the process of apoptosis. So we're going to go over 9.2 today, and then we'll be done. So we're going to talk about the chromosome. So the chromosome is a single double helix. You have one double helix, and it's DNA. Okay? It's composed of more than 50% protein. So we don't realize that a lot. We always think it's just DNA. Like bases that look like a ladder, a twisted ladder. But that ladder has to be wound up around something. And those somethings are proteins called histones. And there's all the different types of structural histones that are in our chromosomes. So human cells contain at least 2 meters of DNA. Do y'all know how crazy that is? That's 6 feet. I think I'm right. You can write me on Schoology if I'm not right. 1 meter is 3 feet. 6 feet of DNA in one cell. We can't even see a cell with our eyes. That's nuts to me. But how does it get compacted so much? Well, these things called histones is why. They pack DNA to make it fit into the nucleus. Each histone wrapped in DNA is called a nucleosome. So I would underline histone and I would underline nucleosome. Nucleosomes are then compacted by folding into a zigzag structure. Then those zigzag structures are put into these things or in the pattern of radial loops to form heterochromatin. And then after heterochromatin, it turns into chromosomes. So I'll show you a picture of what this looks like. I know this is a lot, and please don't just look down at your phone, because if you look down at your phone right now, you probably are failing the class or close. So at the very, very top, you see the DNA, the twisted ladder, okay? Then the next thing you see is these little purple bugs. These purple bugs, they look like bugs, they're really not, but these are histones. So DNA is wrapped around like a spoolie around these histones. Then these histones are put in like a zigzag pattern. And then you've got these radial loops down here. And then eventually this turns into heterochromatin, and eventually it goes to a chromosome. And it's just this little X thing. It's just compacted DNA. That's what a chromosome is. That was super short. I didn't realize that was that short. Look at us go. All right, well, that's it. I'll make another recording for the next half of the chapter. I am going to give you participation points for doing your notes today. And I only do that because I'm not there. And in order to make you take notes, this is how I do it. And especially for those at home, I know a lot of you are at home. I do want you to study and to learn as well. So you can bring your notes back to Miss Willis and show her, or you can email them to me. Okay.